Welcome, everyone. My name is Immaculata De Vivo. I am one of the co-directors for science programming here at Radcliffe Institute. And I have to make a comment about the weather, and why wouldn't I? It's just a spectacular day. And we have a speaker from San Diego, so I'd have to give a little bit of credit to our speaker for bringing us some San Diego weather. So today's event, Twins in Space, is part of the Undiscovered Science Series here at Radcliffe. And undiscovered, we mean, to paraphrase my other co-director of science, Alyssa Goodman, is when you are on a path in science and you are addressing a particular question and you think you know what you're going to find, but you find out something else. And Dr. Rana today, in keeping with that theme, will tell us the amazing resilience of the human body, our ability to adapt in very harsh environments, which is space. So with that, I'd like to introduce Dr. Rana. Dr. Rana is an associate professor from UCSD. Her area of expertise is in twins genetics. Prior to NASA studies, she was invited to serve as the PI of several twin studies investigating relative contribution of the environment and genetic influences on cardiovascular-related traits and diseases, as well as cognitive aging. Previous studies investigated the role of gene and environment, especially on the genome. The NASA twin study was a natural progression of her prior work. And more importantly, Brinda, as I like to call her, is a wonderful personal friend, and she actually took me on this journey with her to be part of this NASA project. My lab generated some of the data for this project and for the paper that's coming up in Science. And with that, I'd like to introduce Brinda.
Thank you, Immaculata, and I just want to say it's a great honor to be here and to speak at the Radcliffe Institute. And I heard I'm competing with Joe Kennedy, so thank you for coming here to listen to me. And thank you for Dr. De Vivo and Goodman for inviting me here. So I had the incredible opportunity to work with NASA for the last 5 years. I've learned a lot. And I'm going to talk a little bit about the study and everything, all my experiences with NASA, as much as I can get in 50 minutes or so. So let me start with a little event that occurred last March. So some of you may have seen the article in CNN in which the article implied that Scott Kelly, our astronaut, went up to space for a year and came back mutated. Now, the great thing about being on the West Coast is while you sleep, the entire world hears the news before you do. And by the time I woke up, every journal in the world had said something about Scott Kelly being mutated. So NASA, in its style, this is what I thought of, like, oh my gosh, 7% of the genome is mutated. We're creating rhesus monkeys up there. Well, NASA was like, what just happened? So the staff at Houston Space Center called us up immediately. We had an emergency phone call at Houston, get to the bottom of it. Well, what had happened was, in January of that year, last year, the twin investigators gave a talk at a NASA conference, just a regular grant investigators conference, and of course reporters were there listening to our talks. Well, the Daily Mail reporter was there, and they kind of misinterpreted all our joint talks. And not only were we mutating astronauts, but we were reversing aging processes, and it went relatively under the radar because, it's the Daily Mail. But for some reason, a month and a half later, CNN decides to read this article and pick up where they left off, and that was what happened. So anyway, with a little help of other scientific journals, National Geographic helped us clear up what had happened. So now I'm going to tell you the real story. Okay, so in March of 2015, astronaut Scott Kelly embarked on a one-year mission on board the International Space Shuttle. And this became the longest journey or longest mission a U.S. astronaut had ever taken on in space. Previous missions for U.S. astronauts were a max of 6 months. There had been several astronauts who would take 3 months and 6 months. And together with the Russian cosmonaut Koryenko, this mission was known as the one-year mission. So the one-year mission alone gave NASA opportunities to study how the human body adapts to long-duration space travel, long duration meaning a year, and also to test out some of the countermeasures against the physiological effects of space travels. Countermeasures is a term they use, therapeutic treatments to counter the issues, physiological issues. So NASA's goal is to send astronauts to Mars in the 2030s. And they want their astronauts to come back safe. They like their astronauts. So they need to figure out, they know some of the effects from the last several decades of space travel. They know some of the effects on the body, but what they're afraid of is stuff that they don't know. Not everything is known. So they're starting to conduct studies on human physiology. So some of the stressors of space travel include microgravity, elevated CO2 inside in the space International Space Center, confined spaces, circadian disruption, radiation patterns that are different there than on Earth, isolation from family, dietary limitation. These are all issues that together contribute to physiological issues. One of the biggest issues that astronauts face, well, let's remind you of the 2 movies that you probably saw, and you can kind of remember of all the things that astronauts go through. So one of the biggest issues that astronauts face is this phenomenon of fluid shifts. We have, as humans, evolved on Earth where there is gravity. So on Earth, our fluids are being pulled towards the feet. Our physiology pumps the fluids, the blood, towards the head. And on Earth, we're fine. We get blood flowing, circulating in our body. When you go into a microgravity or a zero-gravity environment, there is no gravity pulling the fluids back to the feet. So fluid shifts upward towards the chest and the head. So what does this feel like? Well, imagine when you were a kid, if you remember back then, and you hung upside down on monkey bars. And that feeling of pressure in your head, that's the feeling that these astronauts have. Now imagine that for a whole year. So needless to say, this causes a lot of issues. Some of the issues are increased intracranial pressure. They get headaches up there. Edema. So if you look at pictures of astronauts in space, you will see that they look puffy. Now this is not, a photographic thing that happens in space. Their faces are actually puffy. Some people say this is a good thing. A lot of women, older women I've given this talk to say, hey, well, that's not so bad, is it? I'm like, well, okay. So, and you'll see our astronaut, Scott Kelly, on the top when he's on Earth, and on the bottom you can see he's kind of puffy. And then do you notice anything different about Scott on the bottom panel? He's wearing glasses. Okay, so glasses, they have a lot of ocular issues. Now, back in the day, astronauts for like fighter pilots, they had perfect eagle eyes, but now they're representative of the normal, intelligent population. A lot of them wear glasses. But even then, when they go up, they have further issues with their eye. Some of these issues are caused by, they get edema in their eye. They get distension in the optic nerve. And then they get this pattern of choroid folds, which this is a normal picture of the retina, and these are the folds here. Now, I've talked to ophthalmologists. This is not something you see on Earth. This is something that happens only because of space travel. So this is a big issue, big enough that it too hit the media, and this was accurately reported a few years ago. Vision changes as an astronaut, big issue with NASA. So collectively, this fluid shifts, intracranial pressure, eye issues, these symptoms are known as spaceflight-associated neuroocular syndrome, or SANS, formerly known as visual impairment, intracranial pressure, VIP. So they like their acronyms there. Another issue is this issue with orthostatic intolerance. So on Earth, we have a pressure gradient in our body. When you go up in space, that pressure gradient changes. We have little muscles that are pumping blood upwards towards the head. But in space, they don't need to do that because it's already the fluids are shifting upwards, and so they get weak. So when the astronauts come down, the body isn't quite ready to pump blood back up to the head. And the astronauts can pass out. And that's why you see when astronauts land, whether or not they are going to have an event, they are carried out of the space shuttle rather than walking out of the space shuttle. Now, not all astronauts have this event. There's a lot of issues. There's variation in how astronauts respond. And there's a lot of issues, environmental and maybe genetic, that contribute to this orthostatic intolerance. Other issues are cardiovascular changes. So on Earth, if you're a middle-aged adult, you may have gone to screen for atherosclerosis. You look at this, look at this carotid intima thickening of your artery. Normal individuals, they don't have thickening. And if you're starting to develop atherosclerosis, you get thickening. Well, these astronauts, as healthy as they are, when they're in space, some of them develop this thickening. So that's another issue as well. They also have musculoskeletal issues, muscle atrophy, bone deconditioning, and this is kind of easy to predict because they're up there, they're without any gravity, and I've been told by the physiologists that even though they do exercise up in space an hour, a couple hours a day, that still doesn't make up for what we do on Earth with gravity, just the daily walking around. That helps keep our muscle and bone tone going. And then there are potential neurocognitive and behavioral changes that need to be studied as well. So let's put this together. We've got musculoskeletal issues, we've got cardiovascular issues, cognitive issues, eye issues. Sound familiar? So all the issues that we face when we age. So then when people kind of talk to me about this twin paradox, well, did Scott Kelly come back younger? Well, one of our twin investigators, Chris Mason, he actually did the calculations, and yeah, by a few, what did he say? 8.6 milliseconds younger, but really, maybe by timeline, but physiologically, I don't think so. So there are standard changes due to space flight. We know fluid shifts happen. It doesn't matter who you are, you go up there, you get fluid shifts. This is also corresponds to decrease in blood volume, blood cell mass, and changes in urinary output. These are all very standard. in all astronauts. But there are some issues that astronauts face that aren't standard. For example, orthostatic intolerance. Like you said, not all astronauts will pass out when they land. Ocular issues. In studies with the 3 months and 6 month flight astronauts, some of them develop the issues, but some astronauts don't. I think about 60% developed it in the shorter duration flights. And then something you probably don't hear about a lot is astronauts also get space sickness. Maybe it's akin to getting car sick or seasick. I don't think it's talked about that much, but they do get it. And so what's causing these differences in response to space flight? The environment, some of the countermeasures they're doing. The environment's not the same. They're going into space, but the pattern of radiation may be different. There may be events up there in the 6 months or the year that we don't know about that are different. And then their genetics, and then the way the genetics, the genome, the DNA, interacts with the environment and to cause this response. So two of the twin investigators, Dr. Smith and Swart, they are the directors of the Nutrition and Biochemistry Lab at Johnson Space Center. They came up with this multi-hit hypothesis, which is kind of akin to the multi-hit hypothesis for cancer. They talk about that the optic issues that the astronauts face, represented here, is a combination of multiple issues. Their genetic makeup in the one-carbon pathway can cause different response to the oxidative stress. That combined with endothelial dysfunction caused by just environment of space travel, interacting to cause variation in their optical health. So NASA, they've been really good with physiological studies. And in fact, if anyone is doing physiology and wants to do an internship at Johnson Space Center, they're a fantastic lab. It's a great place to learn physiology and environmental response to it. But NASA, 5 years ago, needed to integrate new fields, such as omics, system biology, the metagenome, which includes our microbiome, and then also start studying cognitive issues and behavioral issues that the astronauts are going to face. So to do so, they decided to incorporate these studies into the one-year mission with Scott Kelly going up for a year, perfect chance. Now, Scott Kelly, he reminded NASA, he said, hey, I have a twin brother, and he's an astronaut for NASA, in case you didn't know. And maybe this would be a perfect study because my brother Mark, he's retired. So Mark Kelly was an astronaut. He had 50-some days up in space. He retired a little bit earlier because his wife, Senator Gifford, had just survived an assassination attempt. So he retired to take care of her. So Scott and Mark thought, well, hey, we can do a study while he's up in space. He'll be the space twin, and his brother will be the ground control twin. So Scott and Mark joined the other genetically identical organisms up in the International Space Station. There's rodent studies, fly studies, weeds, Arabidopsis. That's being conducted, and now we had a human genetically identical organism, which was great. So NASA put out a proposal call, an RFA, and they selected 10 proposals. And I was fortunate to be one of the PIs that got selected. And my buddies and I actually put in a couple of proposals, and both of them got accepted. So I was involved in 2 of the studies. And I and my team couldn't have done it without the whole team. Groups at UCSD and University of Texas and your own Immaculata De Vivo. We all worked together for the last 5 years on this study. And what is this study? Well, this study included PIs from all over the United States and 1 in Germany. And we didn't really know what we were going to do. And by the way, I just wanted to tell you that this is a huge study. Pick a number in your head about how much this grant was for. It was $100,000, including indirect cost. And fortunately, I was on a K award, so I didn't have to pay my salary. So we got this. It was really exciting, and got a phone call. I just happened to be in my office when the phone call come from Johnson Space Center. Really exciting. But then the letter comes with this embargo notice. This embargo notice saying, you can't tell anybody, and if you do, it will be extremely serious. And I'm like, whoa, like what am I getting myself into? So NASA called us all to Johnson Space Center in April. Said, okay, pick your co-investigator and fly over to Johnson Space Center. Put us all in a room, NASA style. I think they did this a lot in the 60s. You all are going to be a team. You're going to work together, and you're going to conduct the first integrated omic study of a human body in space. Put your brains together and cooperate. So the idea, the hypothesis is that Scott and Mark, being almost genetically identical individuals, the stressors of space would manifest itself on the genome. So the genome, being the actual DNA sequence or the structure, but really it could change the regulation of genes. So we're changing the regulation mechanisms, epigenetic changes, which then leads to changes in transcriptomics, so DNA to RNA, which then the products of genes, which are proteins, which we were working on, antibodies, metabolites, changes in the metagenome, the microbiome, and then ultimately changes in physiology and cognition. N equals 2. A lot of people like to tease me about this N equals 2 study, but I always say, really, it's actually if an N equals 1 twin pair. I do a different twin studies, and one of the twin studies I work with is actually with Boston University. They have 1,200 twins that they've been longitudinally following, so this is kind of funny to do a N equals 2 study. But anyway, what we're trying to do? Well, I put this up. My postdoc mentor published a paper about how important N of 1 trials are in this day of an age of personalized medicine. So anyway, what we're trying to do is we're trying to just capture events. We're trying to capture molecular events that correspond to physiological changes in relationship with space travel, both good and bad, negative and positive effects. And these will provide clues for further studies. And at the same time, we have the Earth control brother, and just to see how do these a normal middle-aged man, how do these molecules change on Earth and the physiology that we're doing? We're doing some pretty amazing physiology on them. So we got together and created a scheme to sample everything we possibly could: urine, blood, fecal samples, cheek swabs on the astronauts, both on the ground before they go up, in flight, and on the ground, and then with the ground twin wherever he happens to be. Now, we were all molecular biologists. None of us had worked for NASA. Well, most of us hadn't worked for NASA. And we've been designing our studies based on Earth. So, oh yeah, we can do lots for you. That we told NASA on that $100,000 with indirect cost. We can do this, this, and this. Just give us a couple vials of blood at 10 time points in space. We'll do stuff for you. Well, NASA said, yeah, no. Number one, remember I talked to you about reduced blood volume. There's a lot of issues up in space collecting blood. Not only that, we weren't the only study on Scott Kelly. There were like 50 other studies, and they wanted their blood. So they had a limit of how much blood they can give. I mean, these poor guys. And then they're the best subject ever because they collect samples for you. So they had to do their own samples. This is Scott collecting samples for us up in space in the International Space Center. It's not easy. He makes it look easy. And then we thought, well, urine's fine, but no, urine, they don't like the urine process because you have to imagine, you cannot let liquid go airborne. I mean, it starts floating around, and then you get a splash of pee in your face. So they have to pee into a vacuum. And so for our 24-hour pooled urine samples, they got to do it in the vacuum, get a little syringe, and collect the samples for us. It's not easy. And then we thought Scott was hard. Mark was on the trail around the United States giving talks on gun control. We never knew where he was, but at least we knew where Scott was. He was up in the International Space Station. So we're like, okay, well, we need this flight plus 10 days samples. Okay, where's Mark now? So we'd have to oh gosh, he's over in New York. Okay, send the phlebotomist in New York and get blood from him because we need to correspond at the same time as Scott. So that was pretty tough too. So and then we had to all share. Imagine doing a genomic study, just genomics. Usually you get, 1 vial, 10 cc's of blood, and you do transcriptomics or epigenetics. No, you get one cc, 1 vial, and you're going to do transcriptomics, epigenetics, proteomics, metabolomics, everything on that 1 cc of vial. So we had to really work together and develop protocols. Not only that, the protocols were, you got to get everything flight certified. The stuff you use here on Earth, you can't just send it up there because it may not withstand space travel. And if you use glass, it could shatter, and then you got shrapnel all over, floating around, cutting the astronauts. So everything has to be flight certified, and some of my stuff didn't get flight certified. I wanted a filter to filter out the epithelial cells from the urine. Didn't get that done in time, so we just let that go. And then I was a little perturbed when the Italians sent their espresso machine up there. I'm like, they got an espresso machine up there, but they couldn't put my filter up there? But that's the way it goes. So the other thing we had to do, the most important thing that day, they put us in the room for 3 days to work together. The most important thing was the name of the mission. We needed a mission name and a patch. Now, this is important to NASA. I mean, imagine at NIH, you got to get a patch for your study. When an astronaut goes up on any mission, they wear a patch on their flight suit. That patch represents their whatever they're doing. And they'll have tons of patches. Like I said, there's 50 studies that they're involved in. So we had to come up with a name and a patch. Well, and what happens with this patch is, this is Mission Control, Johnson Space Center. At the end of the study, the patch, they make a plaque of the patch, and you'll see all over here, there's these are all mission patches on the walls. And they're ceremoniously hung up on the wall at the end of a mission. And they've been doing this since the Apollo missions. So we had to come up with an idea. Unfortunately, And so even the latrine, the mission that introduced the latrine, the P system, they even had a patch. So, I mean, everything has a patch. So we were trying to come up with names. Unfortunately, the Gemini project had been taken. So we came up with a few ideas. I thought Skywalker project would be fun, but copyright issues. So we finally just settled on the twin study, which was great. And we put every molecule on. Every single one of us wanted our molecule on. And if you look really closely, you've got the DNA methylation, you've got every little molecule on here. And then, of course, the names of all the institutions of the PIs, and I'm very proud that UCSD alphabetically ended up in the middle. Okay, so we got the patch done. Then we had to, in year one, figure out how to get the samples to and from. So to get the samples up, we decided to use, SpaceX. That's really what they use. We had different types of experiments we were doing. Some samples needed to be sent back to Earth fresh. Some of them, for my studies, could wait 300 days. So the Russian Soyuz is actually incredibly timely. So we, for some of the transcriptomics and telomerase studies, we timed the blood draws with the schedule of the Soyuz. And it came back in 2 days. From the International Station, it gets to Kazakhstan. The helicopter is there, NASA's helicopter, where it lands. They go pick up the sample. They rush it over to the Johnson Space Center. And then one of our team would be there at Johnson Space Center to process the samples when it got there. And that's usually Fran, who's the first author of the paper. And for less stringent protocols, we just waited until our shuttle landed in the Pacific, picked up by NASA in the Pacific, taken to Johnson Space Center, and then disseminated to all the investigators. So we came up with this amazing protocol. And, you're working with space. Like I said, things go wrong. And things did go wrong. We did have a supply ship that blew up. But when it did blow up, we got this really nice letter from NASA. If you read the letter, it says, "Speaking as a fellow researcher," I haven't memorized this, "I can only imagine how devastated you must be feeling right now on the loss of SpaceX's CRS-7. And it goes on to tell, how saddened we all are. And then it says, "I'm writing to encourage you, in fact, urge you to continue your inquiry." And can you imagine the NIH sending you a letter like that if your experiment blows up? So we persevered. We just lost samples. They lost food. That really sucked for them. And we got samples. We got lots of samples. And this is just a diagram of the samples that we collected at the different time points. I love the way they write things. L minus 173, so launch minus 173 days, or flight day and return. So we collected from the Earth twin mark and space twin at these different time points, and we did physiological measures along with the collections. Physiology also is complicated. Well, one thing is, one of the PIs, Ruben Gore and Bassner, were doing cognitive studies. Now, they're standard cognitive studies, they're done for neurocognitive testing all the time. But you gotta remember, these are highly functioning adults. So they had to develop a cognitive test for the astronauts, special for them, and one that they could administer through mission control. So remember that, the true cognitive test is what they experience every day. This is what it looks like on the Soyuz, all these buttons they have to press to get around, and they're in Russian, so these tests have to represent their abilities. So that was one of the phenotyping measures that was being done. Our studies were looking at omics in conjunction with these physiological changes, cardiovascular and fluid shifts, the optic issues. So we had to do all the phenotyping, and this is just a slide describing a day in the life of a fluid shift subject. And it's a lot of words, but really what I'm trying to get at is these are all the testing that we did on the astronauts, from ultrasounds to MRIs to dilution measures, pre-flight, in-flight, and post-flight on both brothers. Really, really good physiology. And then in-flight, that's the great thing about in-flight is your subjects are doing the studies on themselves. Best subjects ever. So, like I said, we looked at the carotid intima thickening. So the way we do it, the cardiovascular lab at Johnson Space Center has devised a really nice system where they've got a computer, they've coded it for the astronauts, color and letters. And the astronauts can do their own ultrasound. This is Scott doing his ultrasound for us on the International Space Center. And at Johnson Space Center, Mission Control, there's a little booth somewhere, and then they just call out and they say, okay, a little bit to the left, a little bit to the right. Okay, you got it. Okay, click pink A, capture the image. And then I noticed on the wall in the cubicle, they had all these little squidgy balls. I was like, what's with all these little stress balls? They said, well, there's a 5-second delay between mission control and the space station. And every time they get the best image, and they said, okay, the astronaut moves the probe, and they lose the image. And that causes a lot of frustration, and so they throw balls against the wall. But they're really good. This is my image. I got to do this on myself with them talking me through it, except without the 5-second delay. So I was really proud that I did my own ultrasound. IRBs, NASA had not been involved in genomic studies before. And the biggest concern is here with privacy and confidentiality. The entire world knows Scott and Mark Kelly. Everything we publish, we're going to know it's them. Now, they're federal employees, retired, they got the best retirement plan. My dad was a federal employee, and I know what a great retirement plan they have. So they don't have to worry about health insurance and stuff, but we're talking about genetics, and we don't know what's going to happen downstream with their children, their grandchildren. So we have to be very careful what we say about their data. Everything has to be approved through them, and that's how we wrote this institutional board review. So some of the issues, some of the risks, the main risk is, shooting someone off into space. But at a conference, the International Space Station R&D event, they got Scott and Kelly together to talk about our study, and I recorded it. It's a little bit hard to listen to in a big room, so I'm just going to give you the highlights. So this is Scott and Mark Kelly. The way I remember Scott and Mark is Mark has a mustache. So M for Mark, Scott for space. But sometimes Mark shaves his mustache and changes everything. But this is Mark here. So Mark was jokingly said that there's really no risk for me. I'm just shipping blood, urine, and other stuff to my former employer. He says sarcastically, and then his brother starts talking about the cognitive tests. And he said he was more concerned about the embarrassment of doing worse on the cognitive study of his brother. They're very competitive with each other, like many brothers are. And and he agreed. So Mark agreed. He said that was a lot of pressure, but I did it. But I didn't practice. He said he didn't cheat. He said he did it straight up, and he thought he did okay. And then he proudly says that in the debrief, they said I did better than he did. So I'm happy about that. So, so there's some data right there. And then, of course, in Time magazine, Mark is kind of complaining about sending urine, pee, and poop over to us and the embarrassment factor, but they're ready to do it, and for the benefit of humanity. So these are some of the issues with the IRB. And then the IRB, of course, we got it approved through NASA, and we had to get it approved from my university as well. My university was like, they were like, you know what? This project, you're shooting someone into space, this presents more than minimal risk to human subjects. But they just washed their hands to it, and they said, this committee defers to NASA. So now I'm going to talk to you about our study. So we are publishing our study. It was supposed to come. The reason I decided to come in February was it was supposed to be published a week or two ago. I'm not going to point any fingers, but I was at the NASA meeting last week. It was very sparse because the federal employees couldn't be there, and so I cut out the name, the director of the Human Research Project. They were allowed to come, but as citizens of the United States, and they had to put this on their badge, and they couldn't talk about any NASA stuff. So anyway, it didn't get published, but today we signed off on everything, so it should be coming out soon. This is just to show you the integrative studies, all the different protocols we did, physiology and molecular, all the different time points, and subsequent slides, you're going to see green for Earth, blue for the space twin. I'm going to show you some of the data. And this is really data that the astronauts had approved for us to say. I can't say everything, but you'll get an idea of what we've done. So again, we were looking at my particular studies. We're looking at the correlation between molecular events, omics events, and physiological issues. And in particular, we're looking at metabolites and peptides and proteins. So we did an integrative study looking at, if this means anything to some of you, mass spec-based metabolomics, targeted and untargeted, and proteomics. And then we also had some hypotheses that we were studying about mitochondrial dysfunction in space, and along with our collaborator, Immaculata De Vivo, which was done here, we looked at mitochondrial DNA copy number as well. And then I'll show you some data. So basically what we found was, we found that there were changes with spaceflight in some of these molecules. Some patterns we saw were in plasma, blood, or urine, some of these proteins or metabolites were elevated during spaceflight. In some cases, they were decreased when you compare it to pre-flight baseline levels. In most cases, they came back to normal levels. And then we looked at the brother. Now, what was interesting is, in most cases, the brother didn't fluctuate too much, but they did fluctuate. And in some cases, it could fluctuate a little bit more than the flight astronaut. And you can imagine that an astronaut, when they're actually on a mission, they're in a very controlled environment versus a middle-aged man on Earth over a 2-year period. Their diet, maybe they ate a hamburger one day, exercised one day, had a margarita the next. So they fluctuate quite a bit. But overall, the outcome was a comprehensive and integrated view of how the human body responds to spaceflight, and of a middle-aged man on Earth. which is very important, over a 2-year period. So we found changes in urine metabolites that related to adaptations and multiple physiological symptoms, including fluid regulation, vascular remodeling, and musculoskeletal deconditioning. And I'm going to go through some of the more fun ones that Scott and Mark approved for us to talk about before the paper comes out. Let's talk about hydration. So hydration, very important up in space. We observed a particular protein called aquaporin-2 that was elevated in space flight. So what is aquaporin-2? Well, aquaporin-2 is a protein that's found in the kidney collecting duct cell. It's usually inside the cell, but when the body is dehydrated, the hormone signals a receptor on the cell and says, okay, aquaporin-2, go out to the membrane and bring in water instead of eliminating water in the urine. Bring that water into the cell. We're dehydrated. So that's what aquaporin-2 does. Now, in some clinical studies where they've elevated serum sodium levels, they have found a jump in aquaporin-2. And these are clinical studies that have been done. This is not a normal situation. But we found high-level aquaporin-2 in the astronauts. And then we looked at the serum sodium levels of the astronaut. It does jump in space flight, as does osmolality. So they are very dehydrated up there. And it's not surprising because, as you've heard, they have to recycle their water. They're basically recycling their urine and their sweat, and that's what they drink. And then they have to use this crazy system to pee, and no, they don't like going in there, so then they don't drink enough water. So the whole thing is I'm told, is a setup for dehydration. Energy intake. Well, Scott Smith, one of the 10 investigators, he's the director of the nutrition lab, and they've advanced space food quite a bit. They have lots of choices. They're continuing to do studies to make sure that astronauts are getting the right nutrients. They can design their own food. So it's not as bad as it all is, as one may think, but I'm sure it's not as good as eating down on Earth. So the energy intake does decrease. And this is a graph. If you see the blue line represents a decrease in Scott Kelly's body mass compared to pre-flight. And you can see he drops a little bit more than 8% of his body mass throughout the flight period. This is the other one-year mission astronaut. And the purple is historical data of a group of astronauts over a period of time. Green, as you can see, is brother Mark Kelly, the Earth twin, and as a typical male, middle-aged male on Earth, over the 2-year period, he gained weight. So basically, astronauts are losing weight in space, body mass. And it's not surprising because instead of eating their food, they're playing with their food. So we saw some indications of this in the proteomic study. We found, for example, a particular protein called angiotensinogen, which decreased during space flight. Now, this decrease has been, on Earth in other studies, has been correlated with reduction in body weight. Now, this whole system is also involved in fluid regulation. So what it's going to do, the impact, cause, effect, all this stuff has to be worked out. But we are seeing this, and subsequent studies will work out what it all means. Okay, so when we're talking about body mass, I'm going talk a little bit about telomeres. Now, telomeres on a DNA molecule, at the very end, they're caps, and they serve as caps to protect our chromosomes. They're kind of like shoelaces. The analogy is the little plastic tip on your shoelace that keeps it from being frayed. Without it, your DNA starts to degrade. And telomere length has been associated with age, shorter telomeres with older age. So what we saw in the study, no matter how we looked at it, now Susan Bailey was the lead for the telomere studies of this project, but Dr. De Vivo was part of our team where she helped us validate some of the studies. No matter how you looked at it, Scott Kelly in flight had larger telomere length. Whether we looked at the entire leukocyte population, whether we isolated particular cells, like in this case CD4, CD8 cells, in flight, there was an elevation of his telomere length. And this is a graph of a population of cells and the length, the relative telomere length of the cells. And you can see that in flight, the population of the cells have higher telomere length than pre and post. So basically, telomere length was increased. Now, in flight. Now, studies here on Earth have found that an association of telomere lengthening and weight loss. So this may be the reason, and there might be other reasons. Now, the other thing is telomerase. Now, telomerase is the enzyme that helps put these caps on DNA. In support of the data we saw with telomere lengthening in flight, we saw the regulatory machinery that kind of regulates genes responsible. So epigenetic machinery regulates genes, and then transcriptomics measures the activity of the genes. We saw that supported an increase in telomerase activity in flight, and also some of the genes involved in this process. So this is how we integrated a lot of our studies to show that, okay, we saw it at one level, looking at relative telomere length, but then we also saw it at other levels in the transcriptomics and epigenetic studies. So then we can kind of get to what is the mechanism behind what's going on. The other thing which I find very interesting, being in the Department of Psychiatry, is in November of 2016, 6 months after Scott Kelly returned home, there was a spike in the telomerase activity of both brothers. And this corresponded to a very stressful event. Their father had passed away. So our study captured this event at the level of telomerase. So for what it's worth, this study was able to capture events on Earth as well as on space. Okay, and let's look at exercise. Astronauts need to exercise to maintain their body in space. One of the pathways that we looked at was this lactic acid pathway. So going back to basic biochemistry, if you start with pyruvate, you can go through anaerobic or aerobic metabolism. If you go to anaerobic metabolism to produce energy, you go to lactic acid. If you go the other way, you go through a mitochondrial pathway. What we saw was in-flight increase in lactic acid. So the question was, what's going on? Is it simply due to exercise, more exercise up in space? Is there something going on where they're having lactic acid buildup? Or is it a mitochondrial dysfunction? So we did some studies with mitochondrial function, and that's going to be in the paper. But let's look at the simple stuff first. Well, we were interested to see, well, what was going on in this pattern. This is just the trajectory of lactic acid throughout the course of the study. So this is in flight. It's pretty elevated up here. This would have to do with training. Well, we have to get data from NASA, and it's called this MedB data, and tells us everything about the astronauts, but sometimes the data is not complete. And so we kind of experience this with the data for exercise physiology. So we did what every good researcher does. We went to Costco. We bought the autobiography of our subject. We read the book. And he did say that he was exercising quite a bit, 6 days a week for at least a couple hours a day to prevent all this bone loss and muscle atrophy. So he was exercising a lot. But there was an interesting event. There was a dip here. We also read the book, and we found out that he sustained an injury in his leg during this period where he cut down all his exercise. And that manifested, and we could detect that in the decreased lactic acid. So, but then before landing, he needed to boost his exercise so he could deal with all the issues with post-flight physiology. So he started exercising again, and we detected that. So we did capture some of the events of their regular exercise physiology. Cardiovascular, the vascular. We talked about this CIMT, carotid intima thickening. In flight, he did, so this is pre, post, this is the CIMT measurements. Baseline, and the green is Mark Kelly. Elevated in flight. Post-flight, brother's about the same. He is still elevated on return. And this also corresponds to this carotid diameter, artery diameter. You can see that it's increased up here in flight. So we found changes, we found an increase in collagen protein in the urine of the flight samples. Now, collagen, and we hypothesize that this increase in collagen has to do with the fact that collagen is an important protein in the vasculature. It's also in the eye, in the sclera of the eye. And all this remodeling that occurs due to space flight, this could be causing the shift in collagen synthesis and elimination, and that's what we might be seeing. It hadn't quite been reported in other studies, so this was one of the first novel findings of this study. So this is an interesting marker to look at for some of these physiological vascular events. Microbiome, this is Fred Turek's study. The two dominant bacterial groups, Firmicutes and Bacteroidetes, people often study the ratio F to B's. And what they found is that the ratio increased during flight and returned back to normal in the space twin. In Earth, it was relatively unchanged. Okay, so and then the other findings, there's a lot of findings. We got transcriptomics, methylation data, cytokines, physiology, all sorts of blood markers. They're all integrated in our science article, which should be coming out very soon. We're supposed to get the cover. I don't know if this is the cover, but this is kind of a pilot cover that we have. Okay, so in summary, what does our study show? We saw both transient and persistent changes associated with long-duration spaceflight at the cell level, in different cell types, tissues, and in phenotypes. And our study, our N=2 study, will serve as a roadmap for future studies aimed at understanding potential health risks of long-duration spaceflight. NASA is planning on sending more astronauts up in these long-duration 6 months and 1 year missions, and they are looking at, they have had an RFA and everybody applied to do these studies. We don't know whose proposals are accepted. That we'll know in February as well. So hopefully we'll increase our N and do similar studies. But there's ongoing studies. We also do these simulated bed rest, simulated spaceflight studies. And these simulated spaceflight studies are bed rest studies. So bed rest studies, you're eliminating gravity. And then to kind of simulate this fluid shift, the individuals are tilted headward, downward. And they remain in this position for 30 days, 60 days, 70 days. They eat like that, they shower like that, they do everything like that. And so I'm involved in several of these studies. And then they have studies in which they have countermeasures, and the last study I was involved in where they elevated CO2 for 30 days in these subjects, so they were at a higher CO2 level as well. And then some of the studies, they do exercise, and you can see the difference with these countermeasures or testosterone on their bone health and muscle health. So these are the samples that I'm working on, and these studies are a little bit more, if anybody wants to collaborate on studies, I have the samples, and they're more available than the twin study samples are. And of course, the studies like this has, a lot of Earth relevance for individuals who are at bed rest for aging issues or, injuries. So, in fact, one of my colleagues has some of the countermeasures that he's developed for astronauts is being used by our university to help athletes who have injuries as well. So there's a lot of Earth implications from that come out of these NASA studies. So I'm just going to end with, if there are any students here, graduate students, postdocs, undergrads, who are interested in working with NASA, NASA has a great program. You just go on this intern.nasa.gov. They're taking applications for the summer. I think it's due in April. And they're a great, they're a federal government unit, but they are fantastic. They are so efficient. I feel they are. They might not feel like that themselves, but you ask them for anything, it's there, it gets done. I mean, it's amazing. You always can call somebody and get an answer. Even during the furlough, there was someone there. And then if you're a researcher and you're interested in space studies, I encourage you to go to this NSPIRE site. It's not only the human studies, there are educational projects available, educational grants, and then also all the physics and astronomy grants are available here. So I'm just going to end with thanking just my whole group, the 2 studies that I'm involved with, and the overall group, and Scott and Mark Kelly, and just tell you that, we've had a lot of fun together, 5 years together, working together in Houston. Who would have thought Houston could be so fun? Hanging out, doing stuff, and in the course of this project, 2 of our investigators got some top awards. One of the top awards at NASA is the Snoopy Award. The Silver Snoopy Award. Mike Stenger, who was the PI of the physiology on my study, he was given the Silver Snoopy Award, and I was like, what, is that a real thing? I totally thought it was a joke, but then I looked it up and oh my gosh, that's really cool. And then Alan Hargens, of course, gets one of the big awards that NASA has to give. And then just, if you haven't been to NASA, I mean, you watch these movies and they show NASA to look like this amazing place. It is an amazing place, but it doesn't look like this. It's a federal government building. They still have the tiles from, 1950s, 60s, whatever. kind of looks like this, and this is the cardiovascular lab there. But that's it. So thanks for listening.
Thank you, Brinda. That was great. We have a mic in the middle, and please line up behind the mic and ask questions to Brinda.
What was the story about the genetic changes? Were these epigenetic methylations or?
Yeah, that story, that 7% came from transcriptomics. There was a change in flight that just, yeah, that's what it was. RNA, so it was gene expression changes. Yeah, so there was a change. They got that right. They just kind of, put that word mutated in and it scared everybody.
Hi. This was the first thing ever that I listened about space and twin study. And, okay, forget my story. The question that I have is, why, as a control, what was the reason or not the reason why the Earth control was not given the same food as the space control?
Well, we tried, especially for the microbiome. But if you're a retired federal employee, do you want to eat space food for 2 years? We couldn't get him to comply. We tried, can you at least just eat space food for that couple of days before we do your sampling? And the answer was no.
Even if you made the statement that it's for science, still? No, okay, fine. So I feel that it is a bit less controlled for designs where you're looking at microbiome and stuff. Mm-hmm. The relation between the gut bacteria, which would be highly influenced by the food that they are eating. So maybe I think there are controls to normalize with respect to these aspects, like the kind of food and the nutrients that go in.
Yeah, and not just in the microbiome study. The metabolomic study, it impacts that too, because a lot of the metabolomics has to do with what you eat. And we realized that, and we can combine this study with other astronaut studies and my bed rest studies, which are actually more controlled. We know what they ate in the bed rest studies.
Okay, regarding the bed rest studies, what do you think about something which people use on Earth as a rehabilitation procedure? They train some people inside water, right? Mm-hmm. So how is it different from the space to with gravity? And I think in water, the gravity is a bit reduced, right?
Yeah, they do have the buoyancy studies as well. But buoyancy studies are, they're difficult to do it for a long period of time. Okay. So that's why we use the bed rest studies. But the astronauts actually do study in the buoyancy labs.
Oh, okay. Just last question. Okay, just last question. Okay, so the ground control, was he as isolated as the space control?
Oh, absolutely not. He was living his life. And in fact, I joke about him being a normal middle-aged man on Earth. He's not a normal middle-aged man on Earth. He was flying all over the United States.
So that would completely change the epigenetics and the way mRNAs are produced.
That would, and that was part of what we saw, the variation. But, in general, we didn't see that much variation in a lot of the proteins, that I showed you. It was pretty consistent throughout the 2 years.
Hi, my name is Ilaria Sinelli. I am an Associate Fellow of the Aerospace Medical Association and President-elect of the Aerospace Human Factor Association. So I have a couple of questions for you. The first is that I'd like to know your view on personalized countermeasure, because we can't generalize too much from two twins. Well, from a couple of twins. Yeah. And then can you, from your work, can you extract some result for personalized aging therapies for terrestrial population?
Yeah, From this study, like you said, it's n equals 2, and it's just a first study. I think in combination with what we see here, with some of what I saw in the bed rest studies, I did metabolomics also. I can put some of that together, and I think that will contribute more to earth science than what I've showed here.
I was curious whether you have discovered from these many studies whether there's a minimal time you can go into space, have these molecular adjustments, but come back and totally rebound. I'm thinking, in future decades, we're going to have regular people going up into space for vacations. So what's the possibilities there? Is there a minimal time that you can go up and be okay coming back?
Yeah, so I think the next group of studies we're going to do is going to help address that. They're sending 17 astronauts up and doing similar studies, 1-year missions, 6-month missions, and we'll see. I mean, we saw a lot of rebounding for the astronauts. One of the biggest issues is the ocular issues. That may not rebound. And right now, they have to figure out how to prevent that from happening. So eventually, hopefully they'll find some countermeasures against it, and then they can rebound. And one of the directors of the program gave a talk, and he said, it's not what we know that scares us, it's what we don't know. And as they have more 1-year missions, we'll see what other effects we're going to see. This is just one astronaut, what we saw. Females and males may respond differently. There may be issues that are gender specific, age specific.
Thank you.
Look at me. My name is Giovanni Valencia, and I'm wondering, you were talking about the effects that space has on their eyes for the astronauts. For what I understand, female astronauts, they have less effects on their eyes. So I'm wondering if that is correct, if there are any other changes that happens to male astronauts and not females, and if that also means that the first trip to Mars will be all-female mission.
Yeah, I don't think there's enough data yet to know for sure. You did mention that female astronauts have more issues with the orthostatic intolerance right now. So we do know there's gender differences. Maybe certain physiological effects are better in males and worse in females, and vice versa for other ones. So, time will tell.